Yeah. Hello. Good morning, everyone. Please turn on your camera so we can start the lecture. And also don't forget to rename your name in this Zoom meeting so I can recognize the class and also the number. So later when you want to ask a question or want to have an opinion, so you don't need to mention your name and your class, your number and your class, just mention your name. Yeah. So there are still only few people who turn on the cam. Please, for the others, also turn on the camera. Yeah, thank you. So good morning, everyone. So today we will have a lecture, quite a special lecture because we will have a guest lecture from the Thailand, Professor Jongkon Saising, or we can call it later if you want to ask her. We can call her Miss Aum or Bu Aum. So she will give a lecture about the microbial control and symbiosis for the next 2 hours, and also for next week, and also next week. So she did. So I will introduce her to you. She did a PhD in microbiology in Prince Songkla University in Thailand, right on. And also she did her postdoc in Germany. So she was also working with me in the past. So yeah, so we can start the class now. I will give the time to Miss Aum. So please, Aum, you can share your PowerPoint and start the class.
Okay.
Thank you.
Thank you, Pak Arif, for your very nice introduction. And again, this year, because I already gave a lecture also last year. And for this year, it's also my great pleasure to see you all students today. And before I start, so now everyone can see my slide, right? Can you see my slide? Okay, thank you. And before I start my lecture,
yes,
I just would like to show you a bit about where I am and the university that I work now. And this is our university. The name is Mae Phà Luang University. Here, this is Thailand. And is not far from your country, it's not far from Indonesia. And this is the province that I work in, or our university is situated in the north of Thailand and near to the border of Myanmar, Laos. And just show you the environment of the university, that maybe in the future we can, if the COVID-19 is in, and maybe I can travel to you and you can come to me. And this is the environment in the university. Okay. And I will, as Arif told you, I will give you 3 lectures. And today is our first time. So I will talk about the topic of microbial control 1. And the first part is about control of microorganisms. And as you know that we are surrounded by many types of microorganisms, and especially now that we are faced to the COVID-19 problem. And when we live with microorganisms, how can we live with them without causes, without cause any diseases. So we have many methods to control the microorganisms. And before I go to the methods to control of the microorganisms, I just tell you a bit about the factor that influencing the degree of killing. I mean, killing of microorganisms. The first factors that we need to know about the factors influencing the degree of killing is the type of organisms, the type of the germs, because each germs has their own chemical composition, and this different biochemical composition of different type of microorganisms. They have various mechanisms to protect themselves from the killing. For example, if you look at this figure, you can see here many microorganisms. I think all of you already know about the types of microorganisms. We have the virus, both lipid and non-lipid virus. We have the bacteria, the fungi, which is composed of yeast and mold, and the bacteria called mycobacteria. For example, Mycobacterium tuberculosis that cause tuberculosis, and the bacterial spores. And for this one, it's not, it's actually it's like microorganism, but it is like the infectious proteins that can cause the diseases, such as mad cow disease. And when we have many microorganisms, each microorganisms can protect themselves from the killing of, for example, the chemicals or the methods. And from here, the lipid virus, is the least resistant microorganism. And then the virus is more resistant. From this figure, you can see here, prions, which is infectious proteins, is the most resistant for the killing. And I want to focus here, the bacterial endospores. Normally, the normal bacteria, the normal bacteria can resist to the killing, but when they produce the spores inside the cells that we call endospore, the endospores of bacteria, actually it is the most resistant in the microorganisms. And the next factors, the next factor is the number of organisms. Number of organisms. You can see here, if you have many, if you have high number of organisms, you need more time. You need more time to kill the microorganisms. But if you have lower amount of microorganisms, you need shorter time to kill the microorganisms. The conclusion of this figure, just to tell you that high number of microorganisms require longer time to kill them. Okay, and the third factors is the concentration of the agents that you would like to use to kill the microorganisms. You need the proper concentration of the agents. to make sure that that concentration of the agents can inactivate or can kill the target microorganisms. For example, I show you the example of ethanol or ethyl alcohol, or the alcohol that you use now for protect yourself from COVID-19, or from protect yourself also from other microorganisms. Normally, when I was a student, I think that the 95% of the corn ethanol would be the most the most efficient to kill the microorganisms. But in daily life, that we know that we use only 70 to 80%. And with this concentration, the ethanol can kill the microorganism. We don't need to use the 95% of ethanol to kill the target organisms, because 70 or 80% of ethanol is the proper concentration, and it is the most effective concentration to kill them, and also promote, apart from promote safe, also it is a cost-effective practice. With this concentration, it's safe for our body, for our skin, and it's also promote cost-effective. Okay, and the next factor, you need to consider the nature of the surface, the nature of the surface. For example, the nature of skin, the nature of the materials. For example, that materials are made from plastic, made from rubber, made from steel. Okay, and when you know the nature of the materials or the equipment, you can choose the proper agents or the proper chemicals to kill or to disinfect that equipment or that equipment or that nature from the microorganism. For example, the medical instruments, they need or they require proper disinfection or proper sterilization method to reduce or to kill all of the microorganisms from those instruments or from those materials. Okay, and the next one is the contact time. The contact time means the time that the chemicals or the agents contact to your materials or to nature of the surface that you would like to kill or reduce the number of the bacteria. You need the appropriate time, you need the suitable time to kill the microorganism. For example, if you use the chemicals, the alcohol or iodine, you need 1 to 2 minutes, 1 to 2 minutes for the alcohol or iodine to contact or to touch with your equipment, with your instrument, with your surfaces, or even with your skin. And with this duration time, the alcohol and iodine can kill the microorganisms. But for tolerant microorganisms, it means the microorganisms or the germs that can resist to agents. With this kind of microorganisms, you need much longer time. You need much longer time. Okay, and you need to consider to also the temperature of the chemicals that you want to use. Normally, the temperature of the agents or the chemicals that we use to kill the microorganisms is room temperature. The room temperature is about 25 degrees Celsius. And you need to consider about the pH of the chemicals or the agents that you would like to use. All are the factors that influence the degree of the killing, the types of microorganisms that you need to consider. What is the type? Because each type have the mechanism to protect themselves and have the different level of the resistance to the chemicals or the agents. You need to consider the numbers of the organisms, high number of microorganisms. You need longer time to kill them. You need to consider the concentrations of the agents or of the chemicals. Okay, suitable concentration and consider the safe and also cost effective and nature of the surface, different type of the surface. You need different method to kill the microorganisms. You need to consider the contact time, the temperature, and also the pH of the chemicals of the agents or the method that you would like to use to kill the microorganisms. And for the control of microorganisms, I just want you to know a bit about the common terms when we talk about the control of microorganisms. The first term that you normally need to know is sterilization. I will talk I will show you the example, only these 4 terms. The sterilization, the disinfection, sanitation, and antisepsis. All of these 4 terms talk about the process. Sterilization is the process that destroyed or eliminate all microorganisms and bacterial endospores. Because bacterial endospores has the highest level to resist from the destruction or resist from the killing. With this process, they kill all microorganisms and also kill endospores of the bacteria. And when we talk about this infection is the process eliminate most, you can see here, most, not all microorganisms. When you compare to sterilization, most microorganisms or all pathogens, or the microorganisms that can cause the infectious diseases. But with this process, they cannot kill endospore of bacteria. Cannot kill except endospore of bacteria. This infection, this process cannot kill endospore of bacteria. For this term, used for non-living objects. Non-living objects, for example, used with the materials or the equipment. For example, the surgical equipment or materials or plastic materials. And this infection is the term used for the environment. Used for the environment. The next term we call sanitization. Sanitization is the process, that the pathogenic microorganisms are reduced to the safe level. It means not all microorganisms can be killed with this process. Only pathogenic organisms and just reduce to the safe level. This process is used for inanimate objects. It means non-living objects. Used for the materials. And the last term that I show you is antisepsis. Antisepsis is the process that the chemical agents or the chemicals. These chemical agents we call antiseptics. We call antiseptics. We apply the antiseptics to the skin, or the tissue, the skin or the tissue, to eliminate or reduce the number of microorganisms. Okay, sterilization is used to destroy all microorganisms and bacterial endospores. This infection used to destroy most or all pathogens, but they cannot kill bacterial endospores. This infection used in the case of non-living objects and environment. But if you need to reduce the pathogenic microorganisms, the number of pathogenic microorganisms to level, to the safe level in non-living objects, you need sanitization process. And the antisepsis is the process that we apply the chemical agents. The specific term of chemical agents for this process is called antiseptics. We use antiseptics to reduce the number of microorganisms from the skin or our living tissue. Okay, these are 4 common terms in control of microorganisms. And we have many methods to control of microorganisms. And before we start the methods to control the microorganisms, I would like to ask you, could you please tell me in your daily life, please tell me the way that you use to reduce or to kill the microorganisms. Just give me the example, please. I mean, in your daily life. You don't worry about it is correct or not. Just tell me your daily life that which method that you use to reduce or to kill or to destroy the microorganism.
Yeah. So anyone, if you want to, yeah. So many students are raising their hands. Maybe Miss Aum, you can choose one of them or two of them or three of them, up to you.
Okay, the lucky student I choose is, I'm sorry, is my pronunciation is incorrect pronunciation to call your name, Etikaค่ะ. I think your ID is 051.
051, wait.
Okay, Etika.
Or maybe you can choose the one who raised the hand.
Okay, but I cannot see.
You cannot see. So.
Yeah, actually I can see, but now I cannot see the
because you said so maybe I will choose for you. Is it okay, Aum?
Okay, it's okay. I just want the student to get involved in the class.
Yeah. How many students do you want to answer your question?
Okay, just 3 or 4 is enough. Just tell their friends that which method that they think that method is used to reduce the microorganism in their daily life.
Okay, yeah. So I will choose 4. The first one is Arya, and then the second one is Al Fauzi, and then the third one is Nur Awalia, and the fourth one is Hana. So the first one, Arya, please.
Yes, sir. Is my audio audible?
Yeah, we can hear you.
Okay. Thank you for the thank you for the question. I want to answer that, Miss. First of all, my name is Arya Raka. I'm from C class and my ID is 070. So in my daily life, I'm using with I usually wash my hand. I think that is physical method to clean my hands to clear all the microorganism, the bad ones.
Very
So
good.
I think I
you
use
wash your hand, and which one that you use to wash your hand?
I'm using a soap.
Oh, you use soap. Okay. Very good. The next student.
Thank you, Arya. The next one, Al Fauzi.
Okay, thank you for the question. First of all, I'm Alfonso Jacksono from Microbiology A class, and I have ID 021. I want to answer for the physical method that I use for sterilization or daily life is I use high temperature in an autoclave or oven to sterilize the laboratory tools, and the other one in a chemical method. Usually I use a disinfectant or alcohol to kill the microbial growth, and in a daily life I use like disinfectant or soap for my body. Thank you so much.
Okay, very good.
Yeah, next one. Who is the next one? I forgot. Nur Awalia, I guess. Oh.
Yes, thank you for the opportunity. My name is Nur Awalia. In my daily life, I usually use hand sanitizer before and after I leaving from my home, and after I touch items in public places.
Yeah, thank you. The last one, Hana.
Okay, thank you for the opportunity. My name is Hana from A class. My ID number is 032. I usually boiling my bottle for sterilization, and also I'm pretty agree with Arya and Awalia. I also use hand washing before and after go outing, and use hand sanitizer for traveling. Thank you.
Okay, thank you very much. Both in daily life, we use many methods or many ways to protect ourselves from the microorganisms. And you use, for example, soap, alcohol, and also sanitizer. And some students said they use both physical and chemical method. And the instruments in the lab, for example, autoclave and oven or something like that. And for the method that your friends give the examples, so those methods can be divided into two main methods, the physical method and chemical method. The physical methods, include the heat, the filtration, and radiation. These three physical methods, I will tell you in the detail later. For the heat, the first physical method, we use the heat to kill or to destroy the bacteria. When we use the heat, it can be the moist heat or the dry heat. When we say the moist heat, it means you need water. The water is involved in moist heat. With this method, with the moist heat is faster and more effective than the dry heat. And these are the examples of moist heat that we use to destroy the microorganism. Okay, the first method of moist heat we call steam under pressure. Steam under pressure. It means you use the water. With this method, it's like you boil the water. But if you boil the water in normal condition or in normal atmosphere, the temperature of the water is only 100 degrees Celsius. But if you need high temperature, higher than 100 degrees Celsius, you need to set the condition or the atmosphere like higher pressure. With this method, the temperature is 121 degrees Celsius. Under the pressure 15 psi or 15 pounds per inches, and you need 15 to 20 minutes. Under this condition, you can kill all microorganisms, and you can kill also the bacterial endospores. It is sterilization method, kill all microorganisms as well as bacterial endospores. The equipment that we use for the steam under pressure with this condition is called autoclave. And this picture show you the autoclave. and you can put the things that you would like to sterilize into the autoclave. This one also another autoclave. The second moist heat method is boiling. It's boiling. It's like you boil the water. You boil the water. And with this method, the temperature is 100 degrees Celsius. And when you put the things that you would like to kill the microorganisms, like in your daily life, when you cook the food and you boil the food, or you want to clean some stuff, for example, your clothes or your bottles, you just boil the stuffs, and with this temperature, you need to boil the stuff for 10 minutes, 100 degrees Celsius for 10 minutes, and you can kill the bacterial cells. With the boiling, you can kill the bacterial cells, but the bacterial endospore cannot be killed with boiling. With boiling, you can kill only the bacterial cells, but not their endospores. And the next example of moist heat is non-pressure steam. With this method, it's like you boil. It's the actually it's the boiling method. It's boiling method, but with this boiling, you need to boil or to give the temperature to 100 degrees Celsius for 30 minutes. 100 degrees Celsius for 30 minutes. And then after boiling or after steaming, you incubate. You incubate for overnight, for 24 hours. After that, the next day, you boil it again. You boil for 30 minutes again. And after 30 minutes, you incubate for 24 hours again. You need to do like this on 3 consecutive days, or for 3 times or 3 days. Why we need to do like this? We need to steam or boil and we incubate, and we do it again and again for 3 days. If you look at this circle, for example, you would like to kill the bacteria in the culture media, or in the water or something. And for the first time that you boil or you steam with 100 degrees Celsius, and for example, in your water, you have here the bacterial cell, the bacterial cells. The yellow one here, the yellow ones are bacterial endospores. Bacterial endospores. When you boil the first time, when you boil the first time, you can kill the bacterial cells. The bacterial cells are killed with boiling or steaming. But the endospores cannot be killed with this temperature. The bacterial endospores cannot be killed with this temperature. When you incubate it in 24 hours, incubate it in, it's like the normal temperature or in room temperature or in say 37 degrees Celsius temperature. When you incubate for overnight or for 24 hours, these endospores, you see here, this endospore germinate to vegetative cells. This endospore germinate into vegetative cell. They germinate into vegetative cell. And then you boil it again. You boil it again at 100 degrees Celsius. These vegetative cells, these bacterial cells can be killed. Can be killed. And after that, you incubate again to germinate these endospores. After endospores germinate into vegetative cells, they can be killed. With this method, even you use only 100 degrees Celsius, you can kill all of bacteria and also endospores. But you need 3 consecutive days. The method is also called tinderization. It is tinderization. The next, I think the last moist heat that I will give you the example is called pasteurization. The pasteurization is the process that reduce, just reduce, not kill all, reduce the bacterial population, reduce the number of the bacteria in liquid. For example, in milk, in beverage, or in juice, in liquid product. This method destroy the microorganisms that cause spoilage and microorganisms that cause human diseases. They cannot destroy all microorganisms. They destroy only the spoilage microorganism and the microorganisms that cause the diseases in human. With pasteurization, you can see different temperature and different time. For example, if you use the temperature of 63 degrees Celsius for 30 minutes, with this condition. It is called holding method. The holding method. 63 degrees Celsius for 30 minutes to reduce the microorganism. But if you use the temperature around 72 degrees Celsius for 15 seconds. This method is called flat pasteurizing method. And if 140 degrees Celsius is used for only 3 seconds, this method is called ultra high temperature method, or UHT. It is aseptic condition. And if you notice here, you can see that higher temperature, shorter time. If you use around 63 degrees Celsius, you need 30 minutes. If you increase the temperature, you take shorter time. Higher temperature, shorter time to destroy the microorganism that cause the spoilage and cause human diseases. Okay, this is all for the moist heat method. Now you know the autoclaving or steam under pressure. You know the boiling method. You know non-pressure steam method or tinderization, and the pasteurization with different temperature and different time of pasteurization. And we come to the heat called dry heat. With this method, we don't need the water. Water is not involved in dry heat. And with this dry heat method, you also can kill the microorganisms. The example of dry heat method is hot air oven. The hot air oven. This is the hot air oven. The temperature for this method is 160 to 180 degrees Celsius. And you need 2 to 3 hours. I mean, if you use 160 degrees Celsius, you need longer time. You need 3 hours. But if you use 180 degrees Celsius, you need only 2 hours to kill all microorganisms. With this method, you can use for the equipments or the materials that cannot be destroyed by the dry heat. For example, the glasses, the glass materials, or some or the steel, stainless steel material. but you cannot use the oven for the plastic materials or for rubber materials. Okay. The next dry heat is the method called incineration. Incineration. It is the method that burn to ashes. For example, you would like to burn the infectious waste, or infectious living objects. You need to use incinerator. This is the incinerator. With this method, you need 200 degrees Celsius. 200 degrees Celsius, very high temperature to burn the objects into ashes. And then for direct flaming, this is the last dry heat method that I would like to give you the example. And I think some of you, I think not some, maybe if you learn about microbiology, you already used this method, the direct flaming. Direct flaming. Like when you flame the inoculating loop or the needle, and you need to flame the inoculating loop or the needle until this entire wire is red. And with this method, you also can kill all microorganisms. This is the example of dry heat method. You can use hot air oven or the incineration or the direct flaming to kill the microorganism. The next physical method, is called filtration. Filtration is not the method that kill the bacteria, that kill the microorganisms. It is the method that removes the microorganisms in liquid or gases. How we can remove the microorganisms in liquid or gases with this filtration? We need the filter. This is the filter in different size. Different size or different diameter of the filter. And in the filter, there are many pores, with the pore size, of the filter. If you would like to filter the bacteria, you need the filter that have the pore size of 0.25 micrometer. But if you would like to filter the viruses, or some small bacteria, you need the filter that have pore size 0.22 micrometer. And with this filter, and you can see the different size of the filter holder. This filtration method is suitable for heat sensitive liquid. I mean the liquid that sensitive to heat. It means if you use the heat method, some composition in the liquid will be destroyed. So you need the filtration method to remove the organism. The example of heat sensitive liquid is some growth medium, because you cannot use higher temperature, or the toxoids or vaccine, or some pharmaceuticals, some drugs, some antibiotics, or some blood solutions that cannot use the high temperature. Otherwise, the some composition in these kinds of liquids will be destroyed. So you need the filtration. Not only liquid that you can filter the microorganism. You also can filter the microorganisms also in the air. And you can just also filter the filter that we can use to remove the microorganisms in the air is called HEPA filter. HEPA filter, or high efficiency particulate air filter. You look at this figure. In this case, is for liquid. Here, in the upper part, you pour your liquid, for example, water or culture media, You pour into the upper part, and here is the filter. The water or the medium in the upper part will go through the lower part, go and pass through the filter, In the filter here is the expand of the filter. You can see here there are many pores, in the filter. And the microorganism, here the microorganisms, which are bigger than the pore size, cannot go through the pore size, cannot go through the pores, and still they are still stuck over the filter. Then in the upper part, in the lower part here, your liquid, your water or your culture medium there is no microorganism anymore. Okay, and if you incubate this filter, it means you put this filter on the top of the agar medium in the plate. And after incubation, you can notice the growth of the bacteria. And you can notice the colonies of the bacteria. Okay, this is the filtration method used for the liquid. In case of the filtration for the air, this is the example, the equipment called biosafety cabinet. With the biosafety cabinet, the dirty room air will go through the cabinet and then pass the filter. This is the filter. After the air pass the filter, this area, this working area, will be filled with the clean air. And you can work, you can do your microbiological work without the microorganisms, without the contamination of microorganisms. Okay, this is for the filtration method. The next physical method, I think this is the last physical method that I would like to show you about the physical method is the radiation. The radiation. There are two types of radiation that we can use to kill or to reduce the number of microorganisms from the staff. The first radiation is called ionizing radiation. With this kind of radiation, the example of ionizing radiation is X-ray or gamma ray. The X-ray or gamma ray can pass through the microbial molecules. They can pass through the microbial molecules. We use ionizing radiation with the heat-sensitive pharmaceuticals. It means we use with the things that we cannot use the heat to reduce or to kill the microorganisms for those. For example, we use the X-ray and gamma ray with the vitamins, the hormones, vaccines, or antibiotics. And with the food, we also can use the ionizing radiation. We also can use, for example, the gamma ray to reduce the pathogen or microorganisms in the food. That's why we can keep the foods for longer time. And another radiation is non-ionizing radiation. The example of non-ionizing radiation is UV light, the ultraviolet light. The UV light cannot cannot penetrate the liquids or the solids. They cannot penetrate. So we can use the UV light only for reduce the number of microorganisms where the UV direct exposure to. For example, in the surgical room or in laboratory, we use the UV light to reduce the microorganisms that attach to the surface. For example, the surface of the floor, the walls, and the ceiling, and also other surfaces. Only the surface, they cannot pass through any liquid or solid. Okay, and this is all for the physical method. Now you know many physical methods that can be used to destroy or to reduce the microorganisms for the objects. You know the heat method, both moist heat and dry heat. You know the filtration method, which is not kill the microorganisms, just removes the microorganisms from the objects that you want. And also the radiation method, you know the ionizing radiation, the X-ray and gamma ray, and you know the non-ionizing radiation, the UV light, to reduce the number of microorganisms. Okay, and before I go to the next method, the chemical method, do you have any questions about the physical method?
Yeah, anyone who has a question about the physical method for sterilization can raise a hand. So there are 5 students. Do you want all of them, or how many questions do you want to?
Okay, it is okay, because I think we have time until 9 o'clock, right?
Yeah.
Okay.
So I will call one by one, right?
Okay, it's okay.
Yeah. Do you want to answer directly or collect the question?
Oh, directly is also okay, Arif.
Yeah, okay. So the first one is Nathan. So yeah, please, Nathan, you can ask the question.
Okay, thank you, sir. My name is Nathanael Hariwijaya. You can call me Nathan. I'm from class A, and my ID is 010. Maybe I want to ask something about filtration.
Okay.
Previously, it was stated that there are two types or two size of four size filter. Nah, and when using 0.22 micrometer membrane is for screen for viruses, right? So
yes,
if we use that size, is the larger microorganism will be count, like the larger bacteria. So if we just only want to catch, or we want just only to take the viruses, it's how to do that, like that.
Okay, so your question, you ask about
if we just want to catch the viruses but not the larger microorganism, like that, is we must have use selective media or like something like that.
For this method, we just remove the microorganisms from the objects that we want, and we need to use the proper or the suitable pore size of the filter. And your question is, excuse me.
Can I repeat his question?
Okay.
Yeah, his question is how to separate the viruses only without taking out the microorganism, like without taking out the bacteria. So he wants to separate the viruses only without taking out the bacteria, for example. Is it possible using the pore size 0.22 micrometer?
No, because with this pore size, so we can get both virus and bacteria, because we cannot separate them with the, we cannot separate virus or bacteria. Only use when we use only the filter. But I think, but I'm not sure if you need for the virus, maybe we need the other method to separate the virus. I don't know.
Okay.
You have any idea?
So we cannot separate the bacteria. I mean, you cannot take out the viruses only without taking out the bacteria with the filter of this pore size, right?
Yes, with you can see, if you can see here, because we use the how can I say? If we use the pore size here, and if pore size 0.22, so both virus and bacteria can start in the filter.
Okay. So to separate the viruses.
Okay. I got it, I think. Maybe if you want to separate virus and bacteria, maybe you use 0.25 first to get only the bacteria, because the virus will go pass through this bigger pore size, and then you separate again with the 0.22, and now you can get only the virus onto the filter, I think.
Okay, yeah, thank you. How is it, Nathan? You get the answer?
Yes, it's clear, sir. Thank you, Miss and Mr.
Welcome.
Yeah, thank you. The next one is Ritter. You can ask the question.
Okay, thank you for the opportunity, sir. And ma'am, my name is Ritter. I come from class C microbiology, and my student number is 0.079. And here I want to ask about the pasteurization techniques, especially in ultra high temperature techniques, because in Indonesia we have a brand of packaged milk that use UHT, ultra high temperature pasteurization techniques. This thing make me confused because protein is thermolabile and ultra high temperature require very high temperature. Is it the protein in milk during the pasteurization techniques not damaged? Thank you.
I mean, you mean you worry about the nutrition, right?
Yes, something like that.
You see here, this is the reason why they use very high temperature, but you can notice here, very short time, only 3 seconds. We just count 1, 2, 3, and it's done. So with this high temperature, but very short time here, the protein or the nutrition cannot be, how can I say, destroyed or cannot disappear. And I think you don't worry about the nutrition with this method. But for me, if you compare when you drink milk, and when you compare between the method that they use, for example, the pasteurized milk that they use only 63 degrees Celsius for 10 minutes, just only for my opinion or my experience, I feel that the milk that pasteurized with this method is more delicious than the milk like in with the UHT temperature, because the temperature is lower. That's why the some taste is like, I mean, maybe some fat or some nutrition is still I mean the milk still more tasty than we use the UHT method. But with this temperature and very short time here, don't worry about the destroy of the protein or the nutrition, I think.
Yeah, thank you. How is it, Rita? Do you get the question? Do you get the answer?
Okay, thank you, Miss and Mr. Very clear.
Yeah, thank you very much. The next one is Alpha. Please, you can ask your question.
Yes, thank you, sir, for the opportunities. Let me introduce myself. My name is Alpha. I'm come from Class A Microbiology. I want to ask about ionizing radiation. What do you think, ma'am, about the use of ionizing radiation for sterilization of food? Because the effect or radioactive substance caused are harmful or not for humans, because there are such information say that it is dangerous and some are not. What do you think about this? Thank you.
For me, me myself, I don't care much about that because the X-ray and gamma ray, I think it's not radio exactly radio isotope. It's just the ray that we use to kill the microorganisms. Because I'm not sure until now the report about the use X-ray and gamma ray about, I mean, when we eat the radiation food or something like that, and you worry about the cancer or it affect our health, right?
Yes, I think they are worried about that.
Yeah, but for me, I think I don't care about this, because for me, with this method, they use for quite long time, and I don't know about the report that somebody get the cancer or their health after they eat or they consume the radiation food or something like that.
Okay. So there is still no reports about the people who suffers cancer eats the X-ray or gamma ray food regularly, right?
I think like that, because I think it's just like they're afraid of the radiation or something.
Yeah. The X-ray and gamma ray is totally different than the radio isotope or radioactive, right?
It's not radioisotope.
Yeah, thank you. How is it, Alpha? Do you get the answer?
Yes, sir. Thank you, ma'am, sir.
Yeah, thank you. The next one is Danang.
Okay, thank you for the opportunity. So, good morning, everyone. My name is Danang Samudra. I'm from Microbiology Class C, and my student number is 093. Okay, so I want to ask actually about the steam under pressure that we use outside class, especially the destructive process. So, is there any special treatment when we want to destruct between the bacteria and the fungi? Because based on what I learned, and yeah, something like that, that fungi have some HIVA, and the HIVA are easy to spread. Maybe when we want to destruct, the HIVA will stick to the autoclave. So is there any special treatment when we want to destruct between the bacterial and the fungi in some medium? Something like, thank you.
You mean the method that you want to use to destroy both bacteria and fungi, right?
Yes, that's right.
Okay, and in which object? I mean, if you go back to our first here slide, you need to consider the factors, and one of that you need to consider is the nature of the surface. And if you know that which objects that you want, I mean, with object that you want to be disinfected or to be sterilized, and you can choose the method. For example, if your object is the materials that can resist to high temperature, so you can use the autoclave method, because the autoclave method can kill all microorganisms. They can kill the bacteria. If you see here, they can kill the bacteria. The autoclave can kill all of microorganisms. Both you mentioned about also fungi. The fungi and bacteria also can be killed. And with this, with each method, I think you can use for both bacteria and fungi, but it depends on the number of the microorganisms that you want to reduce. If you want to kill all, so you choose autoclave or boiling. The boiling can also kill the fungi, but it cannot kill only the endospore. And every method can kill both bacteria and fungi, but depends on the number that you would like to reduce. Okay. Do I answer your question?
Yeah, I get the answers. Thank you, Ms. Jongkon.
Thank you, Danang. Thank you. I just want to remind you, I think we have like around half an hour. Do you want to continue answering the question or continue the lecture first?
Okay, maybe we continue the lecture first.
Yeah, okay, thank you.
Okay, and the next major method is chemical method. For the chemicals, the chemical agents and disinfectants. Disinfectant is the chemicals used to disinfect inanimate objects. Inanimate objects mean non-living objects. It means the materials, the equipments, or environments. But if you use the term antiseptics, antiseptics is also the chemicals, but this term used for skin and other living tissue. Disinfectants, the chemicals used to disinfect non-living objects. But if for skin or living object or living tissue, we use the term of antiseptics. Both of them are chemicals. And the categories of chemical agents, the category of chemical agents can be divided into four categories. The first category is chemical sterilant. With this group of chemicals, the chemical in this group can kill all microbes, kill all virus, and kill bacterial endospores. The second group is high-level agents. The chemical in this group can kill all microbes, all viruses, All microbes is mean bacteria and fungi and also viruses, but cannot kill endospore. The third group of chemical agents is intermediate level agents. The chemical in this group kill most microbes and viruses and not kill endospores. And the last group is low level agents. The chemicals kill some bacteria, some fungi, and some viruses, and not kill endospore. This slide just want to show you that the chemicals can be divided into 4 groups. The sterilant, high level, intermediate, and low level agents with different potential to kill the microorganisms and bacterial endospores. And these are the chemicals. That we use for the objects. The first chemical that I will give you the example is alcohol. The alcohol, everybody knows alcohol. The alcohol can denature protein, denature protein of microorganism, and they can dissolve the lipid, especially they disrupt the membranes because the lipid is the component or the composition of the membranes of the pathogen. Alcohol is effective against most bacteria, fungi, and viruses. The alcohol is most effective when diluted in water. The reason to be diluted in water is the prevention of evaporation. So 70% of alcohol is more effective than 95% alcohol or ethanol for controlling the microorganisms. Because if you use 95% ethanol, the ethanol will evaporate. So the contact time to the microorganisms is short. So we need to dilute them with the water to be 70% or even 75 or 80% to prevent the evaporation. And then the alcohol can kill the microorganisms. We can use the alcohol to be the disinfectants for the instrument and the surface to disinfect the surface. And we can use as antiseptic for our skin that you rub your hand with the ethanol. The limitation or the limitation of the alcohol is the skin irritation. When you use alcohol for a long time, you will feel that your skin is dry. It is because of the skin irritation from the ethanol. The next example of chemical is the chemical in the group of halogen. For example, chlorine and chlorine compound. The chlorine react with water to form hypochlorous acid, or the chlorine gas. The chlorine can use in the water, the swimming pool, or in the municipal water supply, because chlorine affects most bacteria, fungi, and virus. And with the high concentration of the chlorine, high concentration of the chlorine, they have the effect like sporocidal effect. Cidal means kill. Sporo is from endospore. The high concentration of chlorine can kill the bacterial endospore. So we call sporocidal effect. The chlorine, like sodium hypochlorite, we can use as surface disinfection. We can disinfect the microorganism from the surface using sodium hypochlorite. Okay. Aldehyde, aldehyde also the chemicals that we use to disinfect the microorganisms from the objects. The aldehyde reacts with the functional group in protein and nucleic acid, which is the structure and the component of the microorganism. The aldehyde, formaldehyde, for example, formalin, can use as disinfectants. In the surgical instrument, we can use the surgical instrument soak in the formalin or formaldehyde, or can use in the isolation room, and used in the dialysis equipments. The 2% glutaraldehyde also used as disinfectants in medical instruments. The aldehyde can kill microorganisms, and they also can kill endospores of the bacteria. But however, the aldehyde in high concentration is toxic to human. So this is the limitation of the aldehyde. The next one, the next example is heavy metal. For example, silver nitrate. The silver nitrate can precipitate the proteins of the microorganisms. The example to use silver nitrate is the 1% solution of silver nitrate in the eye drop. We drop 1% silver nitrate to the eyes of the baby. When the woman give a birth of the baby, and immediately the baby will be dropped with the silver nitrate to protect the eyes of the baby from the sexual transmitted disease, for example, syphilis or gonorrhea from the mother. And the mercury chloride. The mercury chloride is one of the example of heavy metal. These chemicals lies the cell membrane of the microorganism, and we can use as the disinfectant to the object. But the mercury chloride also toxic at high concentration. The next one is phenol and phenol derivatives. For example, the chemical called triclosan. The triclosan denature the protein and disrupt cell membrane of microorganism. They can apply like disinfectants and also antiseptics used for the non-living object and also for our skin. The limitation of the phenol is the bad odor, the disagreeable odor. Their odor is not good. And the chlorhexidine, which is the combination of chlorine and phenol. And we can use as the antiseptics, as the wash clothes with the detergents. The oxidizing agents also can be used as the antiseptics. For example, 3% hydrogen peroxide we drop onto the wounds to kill the microorganisms in the wounds. And you can notice the bubbles here. And the potassium permanganate that we can use to clean our fruit or our vegetable. The next one is gentian violet. The gentian violet, we use 1% gentian violet for the treatment of the oral tract. The oral tract means the infection that is from the fungi, the fungus. And 3% boric acid can use as the eye wash. And one oxidizing agent that interesting is ethylene oxide. Actually, ethylene oxide is the gas, but this gas can use for sterilization of the instruments in the plastic or heat sensitive medical devices. We put the objects into the equipment and we release the ethylene oxide. The ethylene oxide can kill all microorganisms and kill bacterial endospore. Okay, and the last one I think is the soap and detergent. When we wash our hand with the soap also can remove the microorganisms from our hands because of the pH. pH 8, some microorganisms cannot grow in this kind of pH, and they act as the surfactants and can emulsify and solubilize the particles and reduce the surface tension as the surface active agents and can remove the microorganisms from our hand and from our body. And also the detergents, and detergents also act as the strong surfactants. Then they affect the membrane of the microorganisms, and then the cell is lysed by using the detergents. And this is the example of detergents. For example, in the group of quaternary ammonium compound, we use the detergents, both sanitizing agents or skin antiseptics, and also use as disinfectants, like we use for clean the contact lens, and also to clean the walls and the floor in the hospital to remove the microorganisms. Okay, and this all are the chemical method that now I think you know many kind of chemicals that can be used as surfactants for non-living object and antiseptics for skin and living tissues. Okay, and I think the lecture today is finished with the two methods of physical and chemical methods. If you have any questions, you can maybe tell Pa Arif, and then we will answer the question.
Yeah, thank you for the very nice lecture, very nice presentation. So anyone, if you have any question, you can use the feature of raise hand. So the first one is Stephanie. Yeah, please, you can ask your question.
Good morning, ma'am. My name is Stephanie Christina Wahyudi from A class with student number 025. I have a question. Can we use phenol compounds from plants as chemical to control microorganisms?
Okay, of course, you can also use the compound from plants, because in the plants there are many active compounds that can inhibit or also can kill the microorganisms. And actually, it's not only from plants, from other natural products that contain various active compounds you also can use.
Yeah, thank you. So you get the answer, Stephanie?
Yes, thank you, ma'am, for the answer.
Welcome.
Yeah, thank you. The next one is Firda.
Okay, thank you for this opportunity. Before that, I want to introduce myself. My name is Firda from class C with student number 098. Actually, I want to ask about part one, the factor rate of killing. Yes. As already explained, that there are some organisms that have a high tolerance for time, so it takes a lot of time to be killed. And my question is, why do some microorganisms take longer to kill, and what are examples of this microorganism? Thank you.
Could you please ask the question again?
Okay. As already explained, that there are some microorganisms that have a high tolerance for time, so it takes a lot time to be killed. My question is, why do some microorganisms take longer to kill, and what are examples of this microorganism?
Okay, and because the microorganism, the biochemicals of the microorganism is different. That's why they need they can resist to the method or the way that can be killed. For example, if you look at the virus, when I compare, okay, the virus here, the lipid virus. I think, as you know now, COVID-19 is lipid virus, right? Because and with this kind of lipid virus, they can be more easier than the lipid virus or other microorganisms. You need the ethanol, 70% ethanol, so you can kill some virus. But in the case of non-lipid virus, which is more resistant, if you use the ethanol, you need the longer time, or maybe the ethanol is not effective, isn't effective, not enough for non-lipid virus, and you can change to, in the case of other chemicals. For example, you need to use the sodium hypochlorite to kill the non-lipid virus. And with this, you also need the longer time. And also here, when you compare bacteria and fungi, I give you the example in the lab when we do the experiment about the fungi. We cannot, sometimes we cannot use only ethanol to clean the surface, to clean our bench, or also to clean the table. We need sodium hypochlorite to kill the fungi. And with this, we also need the longer time to kill this kind of microorganisms.
Yeah, thank you, for the answer. Do you get the answer, Firda?
Yes, thank you. Very clear.
Yeah, thank you very much for the question also. So is there any other question? You can raise your hand. Or if you have a question, but you don't know how to explain it in English, you can also ask in Indonesian language, and then I will ask Aum in English. I will translate it to you. So no worries. You can also ask in Indonesian language. Yeah, please, Al-Fauzi, you can ask your question. Al-Fauzi, do you hear my voice? I think he has a problem with the connection, I guess. Is there any other who has question? You can raise your hand. So yeah. So for Miss Aum, there will be another 2 lectures, right? The next one for the next week, what is the topic, Aum?
I think the topic is about the antibiotics and the mechanism of action of antibiotics, Arif.
Okay, so the next week topic is about the mode of action of some antibiotics, right? And how about the last lecture next 2 weeks?
The last lecture is like the natural products that we use to use for the microorganisms.
Okay, yeah. So now, Al-Fauzi, can you give your answer? Can you ask your question?
Yeah, yes, sir. I'm so sorry before I turn off my camera because I have a bad connection issues. And here I'm Alfauzi from Microbiology A with the ID number 021. I want to ask about the herbal sterilization with the phenolic or flavonoid. Is that really effective when we compare with the other chemical like alcohol or something like that, or we have a specific compounds that use for phenolic or flavonoid sterilization? Thank you so much.
Your question is?
So his question is about the sterilization using phenolic or flavonoid compounds from the plants, I guess, right, Al-Fauzi?
Yes, sir. What the specific tools are compounds that use with flavonoid or phenolic sterilization compounds?
Yeah, how to use it, how to use the phenolic and flavonoid compound from the plant, like in what kind of sterilization, or sterilization in glassware, or do you have any examples?
The use of the compounds from the plant to control of
I guess.
Okay. It depends on the objects that you would like to use. It means, but you need to like put this active ingredient into, for example, if you would like to, I give you the example. Some compounds can use as it's like antibacterial agents for treatment of the diseases, but some compound you need to add, for example, into the soap or something, maybe but it's like just used to control the pathogens or the microorganisms.
Yeah, so the use of the active compound from plants is usually for controlling infections, right?
I think.
Yeah. Okay. Thank you very much, Aum, for the answer. Al-Fawzi, do you have any follow-up question or is it enough?
Yeah, I think it's enough. Thank you so much.
Yeah, thank you very much. Is there any other question for Ms. Aum regarding the whole topics for today, maybe? No? So I think there is no more question, Aum. So I think the lecture today, we can close it, or maybe you can give a closing statement about the topic for today.
Okay, and I think from the lecture today, I think everybody know about the way to control the microorganisms that you can use for your study and also in your daily life. And if you have any more questions, maybe you can also contact me directly. I will give Arif my email, and you can also write the email to me, or we also if you also not only for this topic but also for others topic and for other of your interesting.
Yeah, thank you very much, Aum, for the statement, for the closing statement. So we can, yeah.
And I also would like to say thank you to Arif and also your students, because they are really good students, active and concentrate to my lecture and asking the question. I really appreciate it. Thank you for your all students.
Yeah, thank you very much, Aum, for the appreciation. So because there is no other question, so we can close the lecture today. And don't forget to also attend for the next lecture at the same time and also on the same day on Monday. So see you next week. Bye. Thank you very much.
Bye. Enjoy your day all.
Yeah, enjoy your day too, Tom. Thank you.